Yes. So now we are moving on to the David Marsden Lecture Award. So this one was created to recognize an outstanding scholar and inspiring neuroscientist in the field of movement disorders. Just to share with the audience how the speakers are selected, as you know, we have an organizing scientific committee for each Congress. It's called CSPC. So the CSPC really comes up with names, and there is a vote for people who can actually meet the criteria. And I'm sure that we all agree that Carly really meets all the criteria for the Stunning Fun Award. And in the case of David Marsden Award, so the society really targets more those who do have an exceptional contribution, which may have resulted in better understanding of the neurobiology of movement disorders. And Andy Singleton, who is very well known of all of you, he meets all these criteria. So Andy is originally from the UK, but he has been working in the US for quite some time, initially at the Mayo Clinic, but he has been for many, many years at the NIH, where he is chief of the neurogenetics department, and more recently, it was created an Alzheimer's and other dementia center. Then he's also chief of that. And in terms of specifically his contributions to our field, he has been involved with the discovery of many genes related to Parkinson's disease and also to some other movement disorders like dystonia, for instance, more than 700 papers. And one of the features that I'm really amazed at your career, Andy, that you have this ability really to network with people from all over the world. So those of you who were present at the opening ceremony yesterday, you may have noticed how really close to our society's heart is our global nature. And that's actually something that I find really admiring and inspiring in your career. So thank you very much for having accepted this award. So please, the podium is yours.
Good morning, and thank you to the President-elect, and the committee for selecting me for this. It's an incredible honor, and thank you for the kind words. So I'm going to be talking today about accelerating and globalizing genetic discovery in Parkinson's disease. And it really is exactly about working with others, collaborating, networking, something that I've really enjoyed in my career. So here are my disclosures. I work for the US government, so I have nothing to disclose. This is what I'm going to talk about. I'm going to talk about four things, really. How far have we come in genetics? Why should we continue? A question I get often is, we found a ton of things over the last 30 years. Why do we need to continue to find things? Hopefully, if I've persuaded you that we should continue, we'll then go to the next part, which is what's left to do, and then how do we get there? I'll also include some recent results at the end, if there's time. So in the context of what's been discovered, back in 1997, the first mutation was described by Mihail Polymeropoulos and Bob Nussbaum, so synuclein mutations. Carly's already talked about that. There are now around a dozen or maybe a little more genes that contain mutations that cause disease. So if you carry them, you have a very high likelihood of disease. In general, those things are pretty rare. LRRK2 is probably the exception, although LRRK2 has reduced penetrance, but in general, they're fairly rare. Anchored in these discoveries of finding monogenic causes of monogenic disease, there have been a couple of discoveries of variants that impart moderate risk for disease. So GBA1 and LRRK2, particularly LRRK2, a couple of LRRK2 variants in the Asian population, those increase risk somewhere in the region of 2 to 5-fold, something like that. A space, so I think most of us geneticists started out in this space, but many of us have moved to this space, which is to try and understand the genetic nature of typical complex disease. And this is really centered around this idea of the common disease, common variant hypothesis, which posits that it's not high-risk variants that drive disease in most people, but it's an accumulation of common low-risk variants that individually impart small amounts of risk. They have an odds ratio of 1.1, 1.2, something like that. But cumulatively, they bring you up to this threshold of disease. So if we look at our current understanding of the genetics of PD in the context of low-risk variants, there are around 90 variants. There are actually about 105 now. There are 15 or so unpublished, and I'll talk about some of those. Individually, they're small risk, but cumulatively, they can be quite large. If you take a population and you look at the individuals in the top 10% strata of risk, they're about 5 times as likely to get disease as those in the bottom 10%. So why continue? We found a ton. We're struggling to understand what those things mean from a functional perspective. Why should we continue to find things? Well, the first answer to that is because there's quite a lot left to be found. We can understand the heritable component of Parkinson's disease, and we think it's around 25%. So if you take an average Parkinson's patient, about 25% of the reason they have disease is due to genetics. That's likely a bit of an underestimate. That number is probably a bit larger, but it's certainly not 100%. So of that 25%, how much do we know the identity of? It's about a third. So we found about a third of this. So there's a ton more to find. And this diagram on the right-hand side shows you how to find the rest, and that is to do ever-increasing studies using larger sample series, doing genome-wide association, and finding more loci. So another question around this idea of why continue is, we have all of these rare variants. We have all of these common variants of small effect size. Why bother looking at those? They only impart small amounts of risk. And this is a really important concept. When we find a new locus, when we find a new a variant, the effect size is about the variant. It's not about the gene that it affects. We're absolutely certain, because of statistical power, that we found something new. So we're sure that there's an effect there. But the variant we find, we're looking at the effect size of the variant, not the effect size of the gene. And the gene can be incredibly important in disease, even if there's a low-risk variant near it. So some examples of this, these 4 genes here, synuclein, LRRK2, VPS13C, and GBA1, these all contain low-risk variants, but they also all contain high-risk variants. So presumably, the same mechanism is being affected to a different extent by low-risk and high-risk variants. So this tells you that when you see a signal, the gene itself is something that's worth studying and understanding in the context of disease pathways. So the point here is that we are trying to modify the pathways implicated by genetics, not necessarily the genetic variant itself. That might change over time. We might start to drug individual alleles, but at the moment we're trying to drug the gene that those alleles affect. There's some real clear data to support this. So this group published a beautiful paper about 2 years ago that centered on this idea of relative success between of drugs that have a mechanism backed up by genetics versus those that don't. So it's this RS metric here. And they showed that if you develop a therapeutic with genetic evidence backing it up, it's about twice as likely to succeed as one without. They've gone into this in more detail in this really recent paper, and I just want to draw your attention to the plot that's focused on odds ratio. And this shows that the effect size of the variant that implicates a gene is irrelevant. As long as there's a sure association there, it can be a small effect. That gene is critical in the disease pathway and really adds great evidence to therapeutic development. So when do we stop? In the context of understanding biology, when we reach biological saturation. So when new genes that we identify tell us nothing more about the biology of disease. Of course, we're not looking in a one-dimensional way. We're not just looking at disease risk. We're looking at all sorts of things like genetics of pathology, genetics of progression, age of onset, response to treatment. So we need to further explore the genetics of those facets of disease. How far could we go? So the latest genome-wide association studies in Parkinson's disease have somewhere in the region of about 40 or 50,000 cases, something like that. A similar trait, height, has a similar amount of heritability. The heritability here is a little larger, about 40%, is one that's been interrogated genetically to a huge extent because, of course, it's a phenotype we all have. This latest study looked at 5.5 million subjects, found 12,000 risk loci. And this is the kind of power that's required to find pretty much all of the genetic variants underlying a trait. So we've got a really, really long way to go. We've only found 100 in PD. There are likely thousands more risk loci to identify. Also, I wasn't telling the whole truth when I showed you this diagram. This is a diagram of success, right? Lots of new things found. But it's an exclusive diagram because it's really focused in individuals of Northern European ancestry. Really not much work has been done outside of that group, a little bit in individuals of Southeast Asian ancestry, but really not a whole lot. And we simply have to do better. So I firmly believe as a geneticist that genetics is the first step to understanding mechanistic basis of disease, to understanding biology. And I think that we have to understand biology to come up with rational points for therapeutic intervention. I also think that we're moving towards a space of precision medicine, as Carly talked about earlier, where we start to match the therapeutic to the mechanistic flavor of disease in an individual or in a group of individuals. So we simply have to do better at understanding mechanism in diverse populations. Parkinson's disease is clearly a global disease. We need to attack it in a global way, and we need to understand disease in really diverse, in ancestrally diverse populations. So how should we do this? So we started a program 3 years ago. I lead this program with Cornelis Blauwendraut, but this program involves thousands of individuals, and I'll show some of them later. This is the Global Parkinson's Genetics Program, supported by ASAP and the Michael J. Fox Foundation. We're supported through 2029, so we have another 6 years of this program. We're about 3 years in. We have a very simple mission to dramatically expand our understanding of the basis of Parkinson's disease, and to do that in a globally relevant way. Again, making understanding of genetics globally relevant. The path is, so a traditional genetics path, because of resources, was for well-funded labs to go out into the field, collect samples, bring them back, analyze them in their own lab and publish. We have a different path here, which is to grow a global research community and to give that global research community the infrastructure, so the tools, the skills, the expertise to analyze their own data in their own research institutions. The resource that we'll produce is genetic data, so dense genetic data in more than 200,000 individuals, again, from diverse populations around the world. So how do we do this? First, by global collaboration and capacity building. So building research capacity in local environments and promoting local investigators. Applying transformative genetic methods like whole genome sequencing, genome-wide association, long-read sequencing. Really importantly, and probably the most fun part, growing the next generation of leaders. So finding those new investigators in underserved populations and giving the opportunities to grow their own research portfolio and to connect. And then lastly, by democratizing data. So democratizing data is not just sharing data. Sharing data is important, but it is making data available and useful to the worldwide community. So this means sharing it, putting it in a place where you can do computational work. You don't need your own compute cluster. You don't need your own supercomputer, and training people around the world to be able to do data analysis. And the idea here is to create this collective, this self-propagating discovery engine where investigators around the world can ask and answer complex genetic questions of their own populations. This requires an enormous amount of operational support. So we have all of these working groups that work on various things like training and networking, looking at issues related to underrepresented populations, making sure we're compliant with the rules which are different in every single country, doing analysis, disseminating data and code. And these groups include hundreds of investigators around the world who really contribute a huge amount of time and effort to the success of GP2. GP2 is also supporting existing and launching new collections and diversity-focused programs. So supporting programs in India, Central South America, Africa, and East Asia, standing up new programs such as the Black PD study in the US, a program aimed at collecting patients from the VA, so diverse patients from the VA, a program collecting patients from Central Asia, and also one focused on diversity in Australia. These are just 8 examples of many programs that GP2 is standing up and supporting. Slide lag again. Okay, we also build global will and research capacity. So again, it's not enough to just put data out there, but you need to build skills and resources for folks to be able to analyze that data. So we've generated 70 online courses, 70 video tutorials, mainly in data science, but also in things like genetic counseling, clinical research, and some basic research. Those are in more than 100 different languages. We have 700 users around the world, including a third from low and medium income countries. So I think we're really starting to build skills in investigators around the world. Okay, we're also sponsoring formal programs. So GP2 has sponsored PhD programs in Mexico, Brazil, Taiwan, and Chile. We have a master's student in data science who's doing the master's at NIH, but is doing it remotely from the Sudan. We have master's students in clinical neurology from Ghana, Djibouti, China, South Africa, and Peru. Again, increasing skill sets in individuals around the world. We bring investigators together. We've had 4 meetings, 2 virtual, 2 in person. The last one was 3 days ago in Copenhagen, where we had 271 attendees from 60 different countries coming together and talking about genetics and how to solve the issue of diversity in genetics. We've done things to build capacity, like support construction of a neurobiobank, hold virtual and in-person courses on data science in Spain, Mexico, and India. We have a whole host of other opportunities over the next year or so. So given all this structure, how far have we come? There are about 170,000 samples in the onboarding and compliance process. If you go to the AMPD website, you can access genetic data on 35,000 samples right now. It's there, it's available, it's easy to access. We have another 45,000 samples in the pipeline for genome-wide genotyping. We've whole genome sequenced 5,000. As you can see, we have pretty good representation around the world. We can always do better, and we're always looking for additional partners who are interested in getting representation for their particular cohort or country. So I wanted to talk about a couple of findings, recent findings. So again, we're only 3 years into this 10-year program, but I think this is a really beautiful example of the type of finding that can come out of this work. This was published in Lancet Neurology, I think 3 or 4 days ago. And this describes the first African-specific risk variant for Parkinson's disease, and really was quite a surprise, but a beautiful proof of the principle of GP2. So this involved groups from Nigeria, from Lagos, Njideka Okabadeju's group in Lagos, a group in UCL run by May Riesig, the Black PD study in the US, and our group at NIH, as well as investigators all over the US as part of the Black PD study. We brought together samples from Lagos, from the US, and from 23andMe and did a genome-wide association study, about 1,400 cases, something like that. If you were to do a genome-wide association study in 1,400 Northern European ancestry individuals, it would be flat. You really wouldn't see anything. So we were really doing this as an exercise. The way that we do the analysis is we do it in the cloud. So it was an exercise in seeing how this would work, working between UCL, Nigeria, and NIH. So we were incredibly surprised to see this standout hit on chromosome 1. So what we have here is an association over the GBA1 gene. This variant that drives this association is prevalent in African populations, almost entirely absent in other ancestral groups. It confers a really substantial amount of risk. So about 1.5-fold increase in risk if you carry the variant as a heterozygous variant, and almost a 4-fold increase in risk if you carry 2 copies of the variant. This is important because it's a really, really common variant. 50% of Nigerian Parkinson's disease cases carry this variant. So from a population attributable risk perspective, this really accounts for a large proportion of disease risk in this population. And I think opens up this population for the first time for precision-based therapeutic trials. We don't have much disease data in other African populations, but we can look at population databases to see how frequent this variant is. It is very frequent in West Africa. You can see in the Isan and the Yorubans, about 30%. But it is also prevalent in African Caribbeans, the Mende from Sierra Leone, in Gambia, in Kenya. So I think this is going to be a variant that is important across Africa, including into South and Eastern Africa. So I think this is going to be a really, really important and widespread observation. So I've talked about the effect size of the allele. If you look at age onset, carrying one allele, again, 40% of the PD population carries one allele, decreases age of onset 2 to 3 years. 10% of the population carries two alleles. They get disease 4 to 6 years earlier. So a really striking effect. What's the functional effect of this signal? So unlike other GBA signals, it's not a coding variant. It's not a protein coding change. We've done a ton of whole genome sequencing. There's nothing there. We've also looked for structural rearrangements using long-read sequencing. There's nothing there. It seems to be driven by expression. So our risk variant is the #1 variant for changing GBA expression in African Americans. So expression at the GBA locus is very complicated because it's a really complex locus with a pseudogene that complicates things, but expression seems to be at least part of the basis of risk here. So I think this is a new mechanism of disease at the GBA1 locus. It's been an awesome group working on this particular project. I wanted to point out May Rizik from UCL, who's worked on IPDGC Africa, and Jadeki Okobadeju from Lagos, along with Lara, who've worked tirelessly to collect samples, Sarah Bandras and Mary Mercurius from NIH, who've done incredible work and training in this group, as well as all the other investigators who've been involved in IPDGC Africa, Black PD. I wanted to talk about another finding. This one is as yet unpublished. This is work led by another wonderful group, Nacho Mata, Jeannie Fu, Mike Knowles, and Kronos Blauendraut. The last study I talked about looked at an individual underrepresented population and looked for new things. This study looks at several underrepresented populations and brings them together to look for similarities and differences genetically in those populations. So this involved samples from Europe, East Asia, South Asia, and Latin America. Again, this is really early days. We're only 3 years into the project. The first 2 years were operational. We haven't collected a huge amount of samples yet. But this really illustrates that even at these early days, you can find new things. So by combining data across these populations, we were able to identify around 10 new risk loci, so taking us up from 90 to 100, and to fine map a lot of the existing risk loci. So one of the challenges is figuring out what the variant is, and this has really helped us. So there's incredible power in looking across populations to see what's common, but also specifically to see what's different. The genetic structure is different, and there are different genetic risk factors in each of these populations. There's an enormous amount to do in GP2. 200,000 individuals with varied phenotypic information. You can look at all sorts of things, the genetic basis of age of onset, risk, progression, therapeutic response, subtyping of disease, especially with integrating things like SAA. But there's also these emerging and adjacent opportunities, right? We have a consortium of 1,000 or so investigators around the world who are also interested in other things and can reach out to other folks within GP2 and say, hey, I'm interested in imaging. Do you have imaging data? How do we work on this together? So I think these adjacent opportunities will be just as exciting as the core genetic investigation. So my take-home message is that we've come a really long way. I think genetics has been incredibly fruitful. There's a ton more to do. There's a ton more to do specifically in ancestrally diverse and underrepresented populations. And those will give us enormous amounts of information, not only about those populations, but about disease in general. It's a social and scientific imperative for us to research disease in these populations. Combining data across populations gives us incredible power, gives us new insights, and looking specifically in individual populations finds new things. I think the GBA1 finding is one of the most exciting genetic findings in the last 10 years to my mind. So it's incredibly exciting. I think this is just the beginning. We're 3 years into a 10-year project. There's a long, long way to go and lots of discovery to be found. We had our most recent GP2 meeting in Copenhagen, in downtown Copenhagen, on Thursday and Friday. I mentioned 271 investigators from 60 different countries, including an enormous number of trainees. There are 170 trainees in the GP2 network, and it was just an amazingly fun and productive meeting. I'd just like to take a moment to thank the Aligning Science across Parkinson's Initiative, the Michael J. Fox Foundation, for being incredible partners in this initiative. It really takes a village to do this work, and to thank the National Institutes of Health, where I've been for 22 years and have just loved my time there. So with that, I will end, and thank you.
Congratulations. So let's let this.